Okay, you guys. So we have a new combination of students in the class. So tomorrow, I am going to let you pick your own seats. So tomorrow is the first come, first serve, wherever you sit, and whoever you sit by, I'm going to pencil you in on the seating chart. Okay, so that's going on tomorrow. So we are going to continue on with genetics. We left off with chapter 14 with genetics, and chapter 15 is also with genetics. There's some things from chapter 14 that we build upon in chapter 15. But in terms of your folders and so on, if you haven't already done so through up to chapter 14, I would maybe take that stuff out and put it somewhere safe at home so you're not lugging it around. So we're not going to be using any of the chapter 13 and before stuff from here on out. So that's also what you can do. All right, so new seats and cleared out your folders. And then the next thing I want to talk about is that it's a fresh start. It's a new semester. So everybody's starting out with a clean slate. Everybody has an A right now. All right, so that should put a smile on your face. Alrighty, so today we're going to continue or start chapter 15. So you picked up your notes for that. So we're going to start here at 15:1. And as I said, it's a continuation of genetics. So chapter 14 was all about Mendel. Remember, Mendel was the guy who worked with peas and figured out you did the P, F1, F2 generation and figured out certain ratios, and so on. Chapter 15, we're going to talk a little bit about another scientist named Morgan. Morgan is famous for breeding experiments with fruit flies. So Mendel was the pea guy, Morgan is fruit flies, and he discovered some things in addition to what Mendel discovered. All right. So the first section here is about the first little part is a little review about the chromosome movement and meiosis. It's just to remind us of that, but also to talk about how Mendel, when he came up with all of his theories about how things were inherited and genes that are inherited, you get one gene from your mom and one gene from your dad. He did all that without really knowing anything about DNA or genes. The word gene hadn't even been used yet, or even chromosomes. When I drew chromosomes and said, okay, this gene is on chromosome pair number 1, Mendel didn't know all of that at that particular time, which makes what he did even more remarkable. So Morgan is the first guy actually to come up with data that supports that a particular gene is on a particular chromosome, and so to match a gene with a chromosome. So that's kind of where we're going here. So in the early 1900s, different researchers proposed that genes are located on chromosomes. So like chromosome number 3 is made out of a piece of DNA and it has hundreds of genes on it, and those genes are on that chromosome and not other chromosomes. And then it's the behavior of the chromosomes during meiosis that accounts for Mendel's law of segregation and independent assortment. Remember, segregation means that you have pairs of chromosomes. One might have a big R, one might have a little r. They segregate or end up in opposite cells at the end of meiosis, so you only pass on one of every kind to your kids. And then independent assortment is how those chromosomes line up. There's 2 different ways for each one of them to line up during meiosis. So this chromosome theory of inheritance states that Mendelian genes, so the genes that Mendel was talking about, have specific loci or positions on chromosomes. And remember that when a chromosome is made out of a piece of DNA, so you have, let's say this is chromosome number 1, so from right here to right here might be the DNA for a hair color gene, all right, and same thing here, all right, and so they're on a particular chromosome. They have a specific location. And it's the actual chromosomes that segregate and go through independent assortment. So not just the genes, but the chromosomes that the genes are on. So that's what we've already known. All right, so like I said, this first part is a little bit of a review. So what this picture does is review the law of segregation and independent assortment. So here's your P generation. P generation, remember, 2 things have to be true. The parents have opposite traits, and they're homozygous for their trait. So notice here it says yellow round seeds. So they're big R, big R, big Y, big Y. And then green wrinkled seeds, little r, little y, little y. So therefore they are opposite but yet homozygous. So when, who's got the thing? Somebody, do you guys see the reflection over here? That's, yeah, all right, thank you. I'm just going crazy. All right, it looks like somebody's got like a, probably mistakenly doing it, all right? And so what happens is when these guys here go through meiosis, they only can pass on one chromosome type to their one of every kind of chromosome. So one with a big R and one with a big Y, because in each pair they're the same. And so this one over here, since they're little r, little y, little y, little y, are always going to pass on a little r and a little y. So all the F1 generation kids are heterozygous. So these guys right here, this is their genotype, and these guys right here, this is their genotype. They're heterozygous for both traits, and so that's what they're showing you here. So then, when you cross these guys to get your F2, remember that to cross these two, to get your F2, remember when we made a big Punnett square? I'm going to relate the Punnett square to this picture here. So when you fill out your Punnett square, you fill out your Punnett square like this parent here, there's 4 possibilities they could pass on to their kids: a big R and a big Y, a big R and a little y, a little r and a big Y, and a little r and a little y. And then since this parent was the exact same, you had the same 4 possibilities here. All right, and so then you fill in the Punnett square. So this right here, from this to this, is showing you, here are the 4 possibilities at the end, why we have the 4 possibilities. So this parent, who when we cross to get the F2 generation, make their gametes, they remember the chromosomes replicate. So then we see here, so the big R and the little r chromosome, the big Y and the little y chromosome have replicated, and they line up with their pairs in the center of the cell. So that's what we see here. So then they separate and segregate. That's the law of segregation. And in meiosis 2, the homologous pairs segregate, so you have the big R chromosome over on the left and the little r chromosome on the right, and the big Y chromosome on the left and the little y chromosome on the right. And then during meiosis 2, remember that the sister chromatids separate. So you have these two having the combination that they could pass on to the kids of big R and a big Y. That's this one here. Or a little r and a little y. These two here, that's this one here. All right? So those are 2 possibilities. But remember that when they line up, the chromosomes line up, they necessarily don't have to line up like this. They can line up in 2 different ways. So notice here, this chromosome pair, the R's, switched. That's the law of independent assortment, that they are each chromosome lines up independently of the other chromosome pair. So now the R switched, the Y stayed the same. So now when it goes through meiosis 1, the little r goes to the left, and the big R goes to the cell on the right. The Y's stay the same. The big Y goes to the left, and little y goes to the right. So now we have the combination here in meiosis 2 of a little r and a little y. So, or big Y, sorry, little r and a big Y. That's that one. And a big R and a little y, and that's this one here. And so that's your 4 combinations here. That's why we have the 4 combinations. So I want to bridge that here. So when we do this dihybrid cross, and you filled in all these boxes, and you figured out the phenotype ratio, you get a 9:3 to 3:1 ratio, which is what we have here. All right? And so that's the chromosomal basis of inheritance. So all of that is a review. So what we're going to now build upon that is what Morgan did with his fruit flies. So Morgan provided the first solid evidence that a specific gene is located on a specific chromosome. He's an embryologist, but then he began to provide the solid evidence for this. He began breeding fruit flies. All right, so His experiments with fruit flies provided evidence that chromosomes are the location of Mendel's heritable factors. Heritable factors, what we're talking, remember Mendel, he didn't call them genes, now we call them genes. So genes are on chromosomes, part of a chromosome. So gene is part of the DNA, which makes up the chromosome. So, so he worked with fruit flies that are have a lot of the benefits that Mendel had when working with peas. So remember we talked about peas being good genetic organisms because you can pick. I want to take this purple flower plant and cross it with this white flower plant. So you can easily pick. They have lots of kids. They grow pretty quickly, and you can get generation and generation and generation data very quickly. Same kind of thing goes with fruit flies. So fruit flies are not plants, they're animals, and so a little bit different there, but they breed at a high rate. So they breed, they have lots of babies, they lay lots of eggs, you can get lots of data fairly quickly. The whole generation, a generation can be bred every 2 weeks. The whole life cycle is 2 weeks. And so you can breed fruit flies, and so what you do is you pick 2 fruit flies that are a couple of fruit flies. One of the traits we'll look at is red and white-eyed fruit flies. So you can put a red-eyed fruit fly with a white-eyed fruit fly in a container, let them mate for a couple of days, all right? You don't need to leave them there for very long together. All right, then what you want to do then is you take out those fruit flies, those parents, because what will happen is they will have laid eggs. And so then you just wait in this container for those eggs to turn into larvae, which then turn into a fruit fly. And then you can look at their kids, all right, and get the data from their kids. All right, and so that's how you do that. And then you can take 2 of their kids and isolate them and put them in their own container, let them mate for a few days, take them out. So what's left and what hatches is their kids, and you can get that data. And so you can get this very, very quickly. So you get lots of data and lots of eggs and so on. And they're relatively simple in that they only have 4 pairs of chromosomes. So that makes it a little bit easier to study genes on the chromosomes and so on and so forth. All right, so, so Morgan noted what we call wild type, or normal phenotypes that were common in fly populations. So wild type, this word here we need to be familiar with. Wild type means most common. It has nothing to do with dominant or recessive. This is what's the most common trait. So eye color. For fruit flies, the most common color of eyes for fruit flies is red eyes. And so if you ever see fruit flies buzzing around, you can actually see the red-eyed color with your naked eye, right? If you just look at them close enough, they have red eyes, and most of them do. And so he called that the wild type, all right? And then any other phenotype besides red eyes, he called the mutant phenotype. Traits alternative to that wild type. So any other color besides that is the mutant. And why he said that, and why he called it mutant, is that, so here's your wild type. What does that mean? It means that the piece of DNA making up the chromosome that has the gene for eye color. So here's the eye color gene, and let's say in this spot of the DNA for that chromosome, and it happens to, the A's, T's, C's, and G's, the order of those bases, code for red eyes. What he said is that most flies have that eye color, and so it has that gene. But the mutant, white eyes, is a mutant. And so there are flies that have white eyes. Why he called that the mutant phenotype is he said that the red-eyed was probably around first and got mutated, where something changed the order of the bases, the A's, T's, C's, and G's. And so now, in the piece of DNA for the eye color gene, instead of the red gene, we have white. So he said that the red gene was mutated, and now we have different forms of the colors of genes. All right, and so which is why we think that we have any diversity here. That in any of the genes that we have, like why we have big R, little r, is that a mutation occurred, and that's why we have different variants of the gene. All right, so that's why he called it mutant. And so here's a picture of the flies, and so here's the red-eyed fly, and here's the white-eyed fly. You can see them, like I said, with your naked eye, the red and the white, but you can also see them more clearly underneath the microscope. So this is the fly eye with red or white. So then he began to experiment with these flies, and he mated male flies that were white-eyed and female flies that are red-eyed. So the white-eyed is the mutant. And it doesn't mean, like people think mutant and there's something wrong, you know, but it doesn't mean that. It's just that it's a variation different than the wild type. Yes?
For example, this be like our eyes, how they think that blue eyes were a mutant type out of brown eyes, which were the wild type?
Yes.
Okay.
And then female flies with the red eye, those are the wild type. And so, so what he found when he did that, he crossed this male white-eyed fly with the female red-eyed fly, is that all the F1 generation had red eyes. Then he mated 2 of the F1 generations to get the F2 generation, and he saw a 3-to-1 ratio of red to white, 3 red to 1 white eye ratio. What does that tell you? Does it tell you anything? What would you hypothesize that's dominant? Red or white?
Red.
Red. All right, we had red and white crossed. All right, we get all red. We cross 2 reds, and all of a sudden white appears. That should tell you that white is the recessive gene, red is the dominant gene. But what was unique about him and this pattern is he got the same kind of pattern that Mendel did. Like Mendel crossed purple and white flowers, got all purple, crossed 2 of the purples, and he got a 3-to-1 ratio. That's what we see here. But what was unique about his situation, Morgan's situation, is only the males had white eyes. You notice that, wow, I'm not getting any females with white eyes. And so that was different. And so ultimately, he determined that the white eye mutant allele must be located on the X chromosome. The X chromosome, remember for us, for that 23rd pair of chromosomes, we have 22 pairs of autosomes. So we have chromosome pairs all the way up to 22. And then our 23rd pair, we either have 2 X's to make us female, or an X and a Y to make us male. Well, with fruit flies, their gender is determined the exact same way. So female fruit flies only have 4 pairs of chromosomes versus 23 pairs like us. So they have 3 pairs of autosomes that don't determine sex, and then they have a pair of the sex chromosomes, and if you're XX in a fruit fly, you're a female fruit fly, and if you're XY in a fruit fly, you're a male fruit fly. But he said that that this gene for eye color must be is on the X chromosome. Well, what happens then is that you have different amounts of genes for eye color if you're a male versus a female, because females for a fruit fly and us have 2 X's, so females have 2 eye color genes for your fruit fly, versus males have an X and a Y. They only have 1 X chromosome, and so therefore if the gene is on the X chromosome, males only have 1 eye color gene for the male fruit flies. And so let's look at that a little bit more in depth. Question?
If it was only showing in males, why would it not just be transferred on the Y chromosome?
That's a good question. At first, that might think about that, because for sure, without a doubt, that if it's on the Y chromosome, females could never have it. What he found is in his P, F1, and F2 generation, he never got females or with the white eyes, but he found that in randomly mating, sometimes you could get females, and I'm going to talk about why that is. Okay. So that's how he kind of ruled out that it wasn't the Y. So let's look at his findings supported this chromosomal theory of inheritance about chromosomes being passed on, having genes on certain chromosomes being passed on to their kids. So let's look at this here. What this is doing is this picture. These 3 go together, these 3 boxes here, P, F1, F2 generation. These are focusing on the phenotypes, and so what color eyes, it's eye color, and what we got in what the flies looked like. These 3 boxes are focusing on the genotypes and looking at what happens to the genes and how they're passed on in the same cross. All right, so these 3 boxes and these 3 boxes are talking about the same 3 crosses, just looking at 2 different things about those crosses. So if you recall, let's look at the P generation. So the P generation, homozygous, opposite traits. Well, let me remind you about this symbology here. So the circle with kind of like the little plus sign or cross coming down from that. Do you guys remember what that means? That's the female. So we need to know that. We commit to memory that. And then the male is the circle with the arrow coming up to the upper right here. And here's those red-eyed cross, so the red-eyed female crossed with the white-eyed male. Okay, so he did this cross, and notice that the kids, the F1 generation, are all red. So I'm going to write here all red, because yours is not color-coded here, so you can't see here. So in the F1 generation, notice he didn't write red females and red males. He didn't separate males and females, and that's just because all of them are red. So whether or not you're female or male, all of them were red. But then when he crossed two red flies from this generation to get the F2 generation, this is where he got that 3-to-1 ratio. So these three had red, and this one had white. But notice though he separated them out into categories. So these guys were female red eyes, a male red eye, and a male white eye. And so the 3-to-1 ratio followed Mendel's rule, but again, noticing that the white eyes only were in males. So what is going on with that chromosomally? So as I said, he hypothesized that we wrote earlier that he hypothesized that the eye color gene is on the X chromosome. So that's what these three boxes here are going through each one of these crosses, but looking at the chromosomes. And so this female red-eyed fly, he said, has red eyes, and she has two X chromosomes. So here's her two X chromosomes. The male has just one X chromosome in the Y. So the Y doesn't have an eye color gene on it. And then notice here, there's a little line and it says W+. He used a little bit of a different notation. So W+. W+ stands for red eyes. And we could hypothesize that because she has red eyes. But the plus, what the plus means, this is a little bit different. It tells us that it's the wild type. So red eyes is most common. That's what the plus means. So she has two red genes to give her red eye color, two red genes on her X chromosome. The male here, he has an X and a Y. He only has one X chromosome. It has the W. W, he said, stands for white eyes. Now, his notation, this is what he used. And so this fly's genotype is W+. This male's genotype is just a W on the X, and no, he doesn't have a second gene for eye color. What doesn't this notation tell us? What was it? Doesn't tell us male or female. All right. So in order to tell that, we need to know if they have two or one, and it doesn't tell us what, Lucas? Yeah, it doesn't tell us what's dominant or recessive. Like when Mendel used a big R and a little r, we knew what's dominant or recessive. So so Morgan, for whatever reason, did not use that same notation. And so in this case here, when you look at somebody's genotype, we don't know what's dominant or recessive unless we know if they're heterozygous, or if we look at the crosses like here, we could tell from these crosses that red is dominant to white. Because of that, sometimes when we're doing problems, if we have the W's, we could kind of blend Mendel and Morgan a little bit. It doesn't hurt anything if we did a big W and put a plus. All right, so then you can tell what's dominant, what's recessive, and then what the plus sign is. It just so happens that red eyes is dominant in wild type. Necessarily, the wild type isn't always dominant. Okay, so what's common isn't always dominant. Question? Oh. Yeah, like your fingers. Yeah. All right. So, so then when these two mate, all right, and so we get the F1 generation that are all red. Let's look at how chromosomally how we got that. So this female can pass on this X chromosome or this X chromosome. Either way, she's passing on a red gene, X chromosome with a red gene to her kids. And then males, either they make half their sperm with the X chromosome, which has a white gene on it, or half their sperm with the Y chromosome, which doesn't have any eye color gene on it. And so then here you get heterozygous female, but since red is dominant, all the females had red eyes. And the males have an X with the red gene and no red gene or no eye color gene on their Y. So they also have red eyes, which is why we get all red in the F1 generation because of that. Then to get the F2 generation, we take two of these. So literally when you're doing genetics crosses with flies, so you put these two together in the vial, you let them mate, take them out, and then the eggs hatch, and then you have all of them have red eyes. So you take a male and a female from that hatching, so really they're like brothers and sisters maybe of one another, and you put those together and you mate those, and so the female, since all the females are heterozygous, they can either pass on the X chromosome with the red gene or the X chromosome with the white gene. And then the male here, he either passes on the X chromosome with the red gene or the Y chromosome. So now when we get here, we can have one female like this that has two red genes and she's homozygous. Or the other combination with female is that she also is going to have red genes. She gets a red gene and a white gene, but she's heterozygous and red-eyed. Or we can have two different kinds of males. Males always get their Y chromosome from their dad. They always get their X chromosome from their mom. So therefore, mom has a red gene X chromosome, so they could have males that are red eyes or males that are white eyes. And so this is where, when we set a 3-to-1 ratio, this is the 3, and that's the 1, and that's the 1 male there. And so in this particular case, with the genotypes of these offspring, males are most often show a trait that's on the X chromosome because males only have one X chromosome. So whatever is on that X chromosome, that's what shows up in a male. Versus, if you look at here, this female here inherited the same X chromosome that he inherited, but she doesn't have white eyes. Why? Because she inherits a second X chromosome from her father, and therefore can mask that. And so, relating this to humans, one of the traits that we know is on the X chromosome in humans is red-green colorblindness. And so what happens is it's a recessive disease. So any female that doesn't have it has either an X big R and another X big R, or X big R and X little r. But on males either, if they have color blindness, they have an X little r and a Y, and so therefore they're colorblind, or an X big R and a Y. And so therefore, when you survey people who are colorblind, there's overwhelmingly more males that are colorblind than females. And that's the reason why, is because females, you can inherit the X with the little r, but females have a second chance to inherit a second X that has the big R and have normal color vision, versus their brother might inherit the X little r and it's a Y, and so he has color blindness. He doesn't have something to mask it. And so we see that more often in males. Anybody in here colorblind? What?
My dad is, and he has 4 sons, and all of them are not colorblind because of their mom. So
that's right.
So we were wondering about that, and now
Dad's always passed on the Y. So yep,
so since mom is
absolutely.
not colorblind.
Yep. So that worked out for you guys. It is possible to have a female that is colorblind. What must be true, do you think, to have a female that's colorblind? She has to have what? 2 little r's, right? X little r, she to be colorblind. So what needs to be true, if this is her, what must be true about her dad and her mom then?
It must be both colorblind, or one has to have like heterozygous.
Who could be heterozygous? The female, because can dad ever be heterozygous for colorblindness? No. What has to be true about dad to have a daughter that's colorblind?
Yes, we colorblind.
He has to be colorblind. He gives his X chromosome to his daughter, right? So mom has to have an X with a little r. So either mom could be colorblind too, in that case all their kids will be colorblind, Or if mom isn't colorblind, mom at least has to be carrying that. And so the rarity that females have the colorblind gene, which is why we don't see colorblind females that much. Although I met my first one today in the class. All the years I've been teaching, I've been asking classes about people who are colorblind. I met my very first colorblind female. All right. So we're going to look at now in humans a little bit, and we're going to make it flow. We're going to skip to 15.3. It flows better, and come back to 15.2. We'll go ahead and flip to 15.3. Yeah, the most common type of colorblindness is red-green colorblindness, where people have trouble distinguishing between the colors red and green. They look very similar. And I have an interesting story. The woman that I did my student teaching with, her husband is red-green colorblind, but he didn't find out he was red-green colorblind until high school. And this is how he found out. So back in the day, they didn't have whiteboards. So he had a green chalkboard in his classroom, and he was in a math class, he said, and he's sitting there, and the math teacher is doing problems on the board. He's asking the students questions about the problems. The students are raising their hand and answering and stuff. He's sitting there, so everybody seems to know what's going on. He's sitting there, sees his teacher writing on the chalkboard, and doesn't see a thing. He doesn't see anything. Like he said it was like really crazy, like because everybody else seemed to know what was going on, and he sees the teacher writing and doesn't see anything. He thought he was losing his mind. So he stayed after class, and what he found out is that the teacher was writing on this green chalkboard with red chalk, and he's red-green colorblindness, and they blended in together. He couldn't see anything, and so I asked him. I said, "Well, what did you think about like stoplights, traffic lights, like red and stuff?" He said it was the location of what light was lit up. Like he just thought the top one, when the top one lights up, that means we stop. You know, he never thought about it.
What about those, like, I've seen them like once or twice, those triangle ones that like go like red, yellow, green. Like what about those?
There are all different types of color blindness. So red-green is the most common, which is red, but there are different types. There are like complete where people see shades of gray. It's not very common, but so there are different types. I've had students that have
like when people say oh, you're colorblind, what color is this?
All right. So let's look at sex-linked genes. Sex-linked genes are genes on our sex chromosome. So in humans and other animals, there's a chromosomal basis for sex. And we know that sex is an inherited phenotypic characteristic. You have physical characteristics based upon the sex chromosomes and the presence or absence of certain chromosomes. In humans, it's the presence or absence of the Y chromosome. The Y chromosome determines maleness. We'll talk a little bit more about that. There's a particular gene on there called the sex determining region of the Y, or the SRY gene, that determines maleness. So therefore, we'll see that it's the presence of that Y or absence of that Y that actually determines maleness. So it's the X and the Y chromosome for humans and some other mammals. Other animals, though, have different combinations. Not every animal has XX and XY, like us and the fruit flies happen to be that way, but it's different in other types of organisms. So let me give you some examples of other types of organisms that it's different in. So here's your XY system in humans. So here, it kind of looks busy here. Here's the male and here's the female. So male has 44 autosomes, all the chromosomes that don't determine sex, and 2 sex chromosomes, an X or a Y, to give him a total of 46 chromosomes. So when he makes his sperm cells, he gives 22 chromosomes and a 23rd, either an X or a Y, 50-50. females have the 44 autosomes and then the 2 sex chromosomes, XX. When she makes her egg cells, she always gives 22 autosomes, the ones that don't determine sex, and 1 of her 2 X chromosomes, but always an X. So with that egg, whatever sperm fertilizes the egg is how we get males or females. So that's the humans. The insects, like the grasshopper here, has the X0 or XO system, where the female is the same combination as us. So the female has 2 X chromosomes. So notice here there's a different chromosome number. The grasshopper has 24 total chromosomes, 22 autosomes, and 2 sex chromosomes. So they have the combination of 2 X's. Notice here the male, it's not the presence of another Y. Males have 1 X chromosome. So it's only having 1 X chromosome that determines maleness. So grasshoppers only have 1 kind of sex chromosome, and it depends on if you have 2 of them or 1 of them to determine gender. A lot of birds, like our chicken here, has the ZW system, where they just call 1 chromosome the Z chromosome, 1 chromosome a W. Notice here, the females are the ones that are not homologous. So in chickens, the females have the non-homologous. They have a Z and a W chromosome. Versus the males have 2 Z chromosomes. So they're opposite in terms of the homologous pairs that we are. And notice also that the chicken has quite a few chromosomes. They have 78 chromosomes in there. And so that also brings me to a point that different organisms have different chromosome numbers, and there's not really a correlation between the number of chromosomes and the complexity or advancement of the organism. So it doesn't necessarily mean that if you have more chromosomes that the organism is more advanced than another organism. All right, so so a lot of people would argue that we are more complex than chicken. Okay, all right, all right. So I hope that you
kind of under debate still, but
so. Then the last one here is with bees, haploid and diploid system. Actually, females are diploid, males are haploid in bees. Males are an unfertilized egg cell actually divides by mitosis and develops into a male fly, or male bee. And so actually males and bees have no fathers, all right? So it's only the females, only the females that the egg gets fertilized. That's why females are diploid, because you have this egg, sperm comes and fertilizes it, now you've got a diploid cell, it divides by mitosis, and the female bee is diploid. Here, you've got an unfertilized egg cell, never gets fertilized. Guess what? It starts dividing anyway, all right, and develops into a male, all right, and that's how that works. Yes.
I remember last chapter we were talking about conditional mutations of recessive and dominant genes. What happens with honeybees with royal jelly or something? How the bee turns into a queen? Isn't that not determined by just normal sexual reproduction? It's something that happens later on.
Yes, that's exactly. That's a kind of an environmental thing.
Okay, yeah.
Yes.
So that would be an example of that. Okay.
Yes. All right, so sex chromosomes have genes for many characters unrelated to sex. So even though they're called sex chromosomes and they do determine sex, there are all kinds of genes, especially in humans, on the X chromosome. The X chromosome is much larger, has more DNA, and therefore has more genes on it than the small little Y chromosome. All right, so the Y chromosome doesn't have very many genes on it. And so a gene located on either sex chromosome is called a sex-linked gene. More often than not, when you're given a sex-linked gene problem, most of the traits are usually for humans on the X chromosome, just because the X chromosome is so much bigger and has so many more genes on it. And these genes follow specific patterns of inheritance. And I have a poster in the back there of the human genome for all of our types of chromosomes, and it shows you different human genes and which ones are on what chromosome. If you're ever interested in that, it shows you that. Okay, so this gives you 3 examples of crossing traits that are sex-linked. So let's assume that, let's talk about colorblindness and how we denote colorblindness. So instead of just using, they used A's, so I'll use A's here, just like a big A or a little a. When it's a sex-linked gene, you put it on the sex chromosome that it is a part of. So in this case here, if we made an X chromosome that has the big A on it, stands for normal vision. Let's do color blindness here. So this is normal vision. And having an X with the little a, I'll just put CB for colorblind. Notice here that when it's on the sex chromosome, we write the sex chromosome and do a superscript of what the gene is on that sex chromosome. So now for females can have any two combinations of these X's, and then males have an X and a Y. So if we look here at this female here, she is homozygous. She has her two X chromosomes. They both have a normal vision gene, so she has normal vision. The male, though, he only has one X chromosome, and it has a little a on it, so he is colorblind, and he doesn't have a second X chromosome. He has a Y, so he doesn't have a second colorblind gene. So whatever males have on that X chromosome, that's what shows up in them. And so, so now when you do a cross here to figure out what kind of kids that they could have, she could pass on either one of her X's. Either way, it's an X with a big A. He passes on the X, which has the little a, or the Y, and these are the kinds of kids that they could have. So if we look at here, they're female kids. When they have a female, they have an X with a little a and a big A for both of these boxes here. It's impossible for them to have a female that's colorblind. And it's also, if we look at the possible boys that they could have, it's impossible for them to have a boy that's colorblind. Even though dad is colorblind, all right? And so it's impossible for them to have a colorblind kid. If we were to do our phenotypic ratio, like you would look at this, and before, when we looked at crosses, your phenotypic ratio, you'd say all four have normal vision. When we look at sex-linked traits, we separate males and females. So your phenotypic ratio would be 2 normal vision females to 2 normal vision males. So we separate males and females. That's different for sex-linked traits. Notice here we have 2 parents now. Neither one of those have color blindness. So she's heterozygous. So it's like taking one of these here. She's heterozygous and he is not colorblind. But now look at what happens when you cross these guys here. Neither one of them are colorblind, but now they can have a colorblind kid because the males, remember dad always passes on the Y to their kids. Mom is heterozygous, so she could pass on, if they have a boy, that boy has a what % chance of being colorblind if they have a boy? If they had a boy, so I'm holding my baby boy, what's the chance that he would be colorblind? 50%, because I already know he's a boy, all right? You see that? That's a difference than asking, what's the chance of them having a colorblind boy? Do you see the difference there in the wording of the question? All right, and so the chance of them having a colorblind boy, your answer would be 25%, because they haven't had the kid yet. Once they have the kid, you know the gender, that changes things. So you know it's a boy, so it's either going to be a boy like this or a boy like this at 50%. All right, and then this is showing you a colorblind male and a heterozygous female. And this is where, like I was talking about here, where you can get your female that's colorblind. All right, so the dad has to be colorblind and the mom either also has to be colorblind or be carrying that colorblind gene. So this right here is very rare. So most of the time you have in a mating here, one of the parents having the X with the little a in there for you. Most often see it in males. So the first thing you're looking at, if you're looking at a trait, is sex-linked traits generally you see more often in males. So that's the first thing you kind of would look at. All right, so some disorders are caused by recessive alleles on the X chromosome. So we've already talked about color blindness. Another one that we know is on the X chromosome is Duchenne muscular dystrophy. This is a degenerative disease of the muscles, the working of the muscles. And hemophilia. Hemophilia is a blood disorder, blood clotting disorder, where the people who have hemophilia do not make a certain clotting factor, they call it. It's a protein that helps your blood to clot, and so therefore they can risk bleeding to death with injuries if not given that clotting factor. So these are all recessive alleles, which means that people who have these are either, if you're a female, you're X little r, X little r, or if you're a male, you're X little ry to have these. They're all recessive in humans. So what I'd like you to do is kind of on the bottom or in the margin, without looking back on the other classes here, these two bullets here, what I'd like you to do is do these crosses and write out the phenotype ratio. So in the margin somewhere, we're not going on in our notes yet, quite yet. I usually wear my phone that way. So I think it's supposed to go there. I like it. So your heterozygous female should be X, I'll use A's, you could use R's, whatever, X, big A, X little a. Your colorblind male has X with the little a and the Y. So this is how you set that cross up. And when you figure out what kinds of kids that you could have, they could have one normal visioned female, one colorblind female, one normal visioned male, and one colorblind male. So when you separate out the males and females, it's a 1 to 1 to 1 to 1 ratio. All right? And so then in this case here, she's homozygous recessive, so she's colorblind. He's also colorblind, and so automatically we should be able to tell they can't have anything but a colorblind kid. And so this is what the cross looks like here. So you're going to have 2 female colorblind to 2 male colorblind, or you can reduce that to a 1 to 1 ratio. Okay? And so that's how that works. All right. So what I'd like you to do is look at the single sheet of paper that you picked up here. It has some pedigrees on it. And this is what you are going to do for tomorrow. Oh. Yes, that was my way of saying it's homework. Without using the H word. This is what I want you to do. So you have 3 pedigrees on there. you can disregard the meiosis picture there. So you have 3 different pedigrees. Remember a pedigree. What does it tell you? It shows the relationship of people, but the shaded in people are affected with the trait. So, so those are the people affected with the particular trait. What I want you to do is to determine whether or not the trait that's depicted in that pedigree is a trait that is autosomal dominant. So autosomal means it's not on a sex chromosome. Dominant means the trait is dominant. So we talked about Huntington's disease. Huntington's disease is a dominant trait that is a degenerative nerve disorder. And so what happens is that people with Huntington's have a big R something, all right? So because it's a dominant gene, which means that everybody who doesn't have Huntington's would be little r, all right? If the trait that of the people shaded in is autosomal recessive, autosomal again meaning it's not on the sex chromosome, recessive like cystic fibrosis, that means everybody who has the trait would be little r. This would be have cystic fibrosis, meaning that everybody who doesn't shade it in would not be little r. They'd have to have at least one big R. Or the trait could be sex-linked, like we just talked about, like hemophilia or colorblindness. So the people like this would either, if you're females, would have X little r, or if you're a male, you'd have X little ry. So what I want you to do is look at a pedigree, the 3 pedigrees, look at it and make a hypothesis. Do I think that people shaded in have a disease that is autosomal dominant, recessive, or sex-linked? After you make your hypothesis, what I want you to do is test your hypothesis. This is how I want you to do it. Do this with pencil, just in case you make a mistake. So let's say you think the trait is autosomal dominant. So then everybody shaded in, you'd put a big R by them. Everybody shaded in would be big R. Then everybody not shaded in would be little r. And trace the genes through the family and make sure that there's no discrepancies. Let me show you what I mean by a discrepancy. Let's say you had this was part of your pedigree, where these 2 parents had this girl. And let's say your hypothesis was that it was autosomal dominant. That means that she would have to have a big R, which means that since they're not shaded in, they'd have to have a little r, because everybody shaded in would be autosomal dominant. Is it possible for those 2 to have that girl? Absolutely not. That tells you that your hypothesis is wrong. That's what I mean by a discrepancy. So there's 3 pedigrees, and I want you for each one to tell which mode of inheritance it's on, okay? By checking yourself, all right, with the...
So there's one of each.